afternoon one and all, and welcome to this lecture series, which is part of the Festival of Science that we are celebrating. And the theme for this particular lecture series is Swadeshi, Paramparik Innovations and Inventions. And to deliver a talk on this theme, we have a very distinguished scientist today with us, Dr. K. Thangaraj, who's the director of the Center for DNA Fingerprinting and Diagnostics. I would now request Dr. Ahmed Ibrahim to kindly accompany Dr. Thangaraj onto the dais. And I now request Dr. Ahmed Ibrahim to kindly honor the guest with a bouquet. It's my pleasure to introduce Dr. Thangaraj. He's the director of Center for DNA Fingerprinting and Diagnostics at Hyderabad. Prior to this, he was chief scientist and group leader at the Center for Molecular Biology at Hyderabad. And his main research interests are origin of modern humans, cardiovascular diseases, mitochondrial disorders, and male infertility. He has published over 275 research articles in high-profile journals which include The Cell, Lancet, Science, Nature, and Nature Genetics. He is an elected fellow of the Indian Academy of Sciences, Indian National Science Academy, and National Academy of Sciences. He is a recipient of the J.C. Bose Fellowship, Sun Pharma Research Award in Medical Sciences, Raman Research Fellowship, Lifetime Achievement Award, Excellence in Science Award, Distinguished Scientist Award, and many more. He is a board editor for several journals. So I would now request Dr. Thangaraj to please deliver his talk. Sir.
So, good afternoon to all of you. Thank you very much for your kind introduction. Also thank Dr. Paramparik Reddy for inviting me to talk to this audience. I feel very happy that almost all of them are students. So it's a great pleasure for me to be part of this overall meeting. So what I'm going to talk to you is how studying the population genomics is going to help us for better understanding of our health and disease. So what I'll be talking is that, so one of the curiosity is that how we are all originated, right? So any species origin is very important and interesting. Particularly understanding of our own origin is even more curious, right? When you talk about population study, there is no other country in the world to have such a huge human diversity. The slide itself says that even if you look at among ourselves, that how much variations we have in terms of our color, in terms of our hair, in terms of our height, and there are many more, right? So that tells that how much human diversity exists in our country. Another important aspect is that our Indian population are stratified in many ways. Broadly, you can say what is based on our language. So we have 4 major linguistic families. We, as a South Indian, we speak Dravidian language, right? Telugu, Kannada, Malayalam and Tamil. And there's a Indo-European language, people from the north. There's an Austroasiatic, people living in this part of the country speak Austroasiatic language. And the people in northeast and north, one part of the north speak Tibeto-Burman language. Okay, these are the 4 major linguistic families. As we have 4 Dravidian languages, similarly under each category there are several languages. In addition to that, there are some isolated languages, like the people in Andaman and Nicobar Island, they speak Andamanese language. There's a language called Nihalis, and there are many more. They are called as isolated language. And again, under each linguistic families, there are several social stratification. We all know that our population, in terms of social structure, there's a caste system, right? Religious system. Under caste system, we have so-called upper caste, middle caste, lower caste, tribes, primitive tribes, and so on. Okay, so all of them, if you put it, so makes India as one of the very complex country in terms of human diversity. So this is one introduction I wanted to do. Then to understand the population history, there are many tools being used initially. But now genomics is one of the important tools. Right? So when you talk about genome, you all must have studied. Genome is nothing but, in a simple term, is a composition of entire genomic information, or the sequence present in the entire cell, if you wanted to look at only cell. Again, there are 2 different kind of genome. One is the nuclear genome, which comes from the nucleus. In human, we have 46 chromosomes. 23 we inherit from father, 23 from the mother, equally. Right? And in addition to that, there is a mitochondrial genome, which comes from mitochondria. You all know the mitochondria is known as powerhouse of the cell, because all the energy required for all our function comes to the mitochondria. So these are the 2 different genomes, and these genomes are inherited differently. So if you take mitochondrial DNA, that comes from only mother. Okay, the somatic cell, as I mentioned, this is a female karyotype. You must have studied the karyotype, right? Arrangement of the chromosomes. This is a male karyotype. The difference is only the sex chromosomes. In case of female, there will be 2 X chromosomes. In male, 1 X and Y chromosome. This is a chromosome which consists of a gene responsible for making individual as a male. It's called as SRY, sex determining region of the Y chromosome. So this is from the somatic cell. You know that entire body, there's a somatic cell with the 2 set of chromosomes. But in the germ cells, like egg and sperm, it will be off of it. Okay? Off of that. After these 2 pairs, only 1 chromosome will be there. Out of these 2, 1. So this would be in case of egg, this is in case of sperm. But if you talk about mitochondrial genome, that's completely present in the egg, mitochondria, but in the sperm, mitochondria present only the midpiece. At the time of fertilization, it remains outside. Therefore, there's no contribution of paternal, that means the father's mitochondrial genome. So we all inherit mitochondria and mitochondrial genome only from the mother. Okay? So we study these 2 genomes for addressing large number of questions. So initially we used the complete mitochondrial DNA, and we have been studying this for last 25 years to understand Indian population history. So we used the mitochondrial DNA for studying the population called Andaman and Nicobar Islanders. I don't know how many of you are aware, there are large number of islands. in the eastern part of the country, there are 572 small islands. There are 4 tribal groups. They typically look like Africans. They are called Onge, Jarawas, Great Andamanis and Sentinelese. There are 2 tribes called Nicobaris and Champagne. They typically look like Southeast Asian groups. Okay. So we studied the complete mitochondrial DNA by taking their blood. This is a Onge tribe. So this was taken very, very long back. We studied the complete mitochondrial DNA, which is 16.5 kilo base, 16,500 base pair. And we identify the variations which are evolutionarily conserved. And based on the mutation, we assign a specific haplogroup. These are the mutation which is present only in Andamanis. And based on the mutation, one can also calculate the age of the population. By looking at that, we calculated that the Andaman tribes are the first modern human who migrated out of Africa, taking southern coastal route and reached Andaman and Nicobar Islands about 65,000 years back. This is the first genetic study which confirmed that the Andaman tribes are the first modern human who migrated out of Africa, taking southern coastal route and reached Andaman and Nicobar Island about 65,000 years back. Okay. Subsequently, we studied large number of populations. Just I am highlighting the very important findings. Then we used the nuclear genome. Okay, so now after human genome was sequenced, 1 million genetic variations have been identified across the genome. It's called as single nucleotide polymorphism. So we used that and studied large number of Indian groups representing all the 4 major linguistic groups: Dravidian, Indo-European, Austroasiatic, Tibeto-Burman. And also representative social groups from each linguistic groups. And we finally end up with 560,000 single nucleotide polymorphic marker. Then we compare that with worldwide data set of human genome diversity panel and HapMap data set. There are several statistical analyses. So using this, in fact, we reconstructed the Indian population history. This is one of the landmark paper where we have shown in the prehistoric India, there were 2 founding populations. Sorry for that. One is ancestral South India. If you look at this divergence, as I mentioned, Andamanis are the first modern human migrated out of Africa. It's a very early divergence. And while Andamanis were migrating towards Andaman and Nicobar Islands, some groups stayed in southern part of India, whom we call as ancestral South Indians. Okay. And the second wave of migration brought people from Africa towards north, and somewhere in the Middle East, again there was a divergence. One group has gone to Europe and other group has come to northern part of India, whom we call as ancestral North India. So these are the 2 founding populations. And these 2 founding populations, there are several studies subsequently we have shown that how we are linking that Europeans and ancestral North India has some kind of genetic affinities. This is based on the mutation which is common in Europe, Middle East and northern part of India. 2 examples. One is the skin color mutation, which is common across Europeans, Middle Eastern and northern part of India. Then the lactate persistent gene mutation, okay, which helps in digesting the milk sugar efficiently. But that's again common in the northern part of India, Europe and Middle East. Okay, there are a few more examples which I am not going to detail. Then these two groups, ancestral North Indian and ancestral South Indians, have admixed. That's why if you see today, there is no distinct North Indian group, no distinct South Indian groups. We are all admixed. And the many features, including the color, the gradient, if you take extreme north, very light color, extreme south, dark color. In between, we have so many different colors that itself shows that there's a kind of admixture took place. Another complexity in terms of Indian population is there are several migrations happened in the recent past. Some of the historical things which you know, that is the invasion of Alexander's army, then military conquest by Arab and Turk, British colonization. There are few populations who have migrated recently, including Siddhis. There's a place in Hyderabad, there are few Siddhi populations exist. Population also in other part of the countries, which I will tell you in coming slides. Jewish population, Parsis, and many more. Okay, so all of them put together makes India a very complex country. If you look at the origin of these groups, I'll give you very brief examples. For example, if you look at Siddhis, okay, who are in Hyderabad, also in Gujarat and Karnataka. We analyzed all of them. And historically, they have been brought by Portuguese traders and they sold here to work in the army. Okay. That happened about 400 years back. And if you look at their genome, there are three different kind of genome. Autosome, which we inherit from both the parents. Mitochondrial DNA, I mentioned that we all inherit only from the mother. And the Y chromosome, that's only goes from male to other next generation male, grandfather to father, father to son, grandson, it goes like that. And if you look at the African genome, Siddhis and Indians, the proportion of mitochondrial DNA and Y chromosome itself suggests that mostly the males have come and married the Indian woman. So that again is a kind of admixture in this case. Similarly, if you look at Jewish population, they are again in different places, particularly in Kerala, in Mumbai, and also in Calcutta. This is called as structure analysis, again using 1 million genetic markers. So it gives different kind of color based on the genomic similarity. For example, if you look at the South Asians, including India, Pakistan, you have the green color, which is very predominant. That means this kind of genome, more than 50% of genome, is very unique. Right? Then if you look at the Jewish population here, where you find that dark blue color, so that is predominant here, Africa, Middle East, but you can see it's a very, very small proportion here. That means they came here and admixed thoroughly because they have more of the Indian genome than the original Jewish genome. Okay. Then if you look at the Parsis, you know that Parsis are also here. And again, so these are the Parsi populations. So they carry little more their Parsi original genome than Indian genome. Of course, they are also admixed. But if you look at the ancient genome, you know that Harappa is the place where the civilization started. Historically, you all know, you must have studied. Right? So there was a possibility for us to analyze this bone sample. Of course, we collaborated with the large number of archaeologists who have excavated this place and identified the intact bone, skeletal of a woman. So we sequenced the whole genome. So what we come to the conclusion is that when we compare that genome with existing or contemporary population genome, it shows that this is the Indus Valley genome or Harappan genome. It consists of Andamanese related genome, the small proportion, and large proportion have come from Iranian hunter-gatherer. Okay? So that's of more than 10,000 year old. much before the agriculture came into existence. The agriculture started at a place called Fertile Crescent in Iran. Okay, so that genome, if you look at the Iranian farmer, they are the one probably have this agriculture related origin. But this is not Iranian farmers, they are Iranian hunter-gatherer. Okay? But the sample itself is not very old, it's only 3000 BCE, but they consisting of Iranian hunter-gatherer. That means they migrated much before the agriculture came into existence. And we came to concluded that probably because they have a hunter-gatherer's genome, agriculture must have started independently in India. So that's a different kind of conclusion. So what I have been showing so far is that how complex the Indian populations are. Okay? And it's not exaggeration to say India is an ethnic museum because we have collection of large number of population groups. And to give you some idea that how many populations are in India, and you may be surprised to know that in India we have 4,635 anthropologically well-defined population groups. Nowhere in the world will have such a huge human diversity. That includes large number of tribes, primitive tribes, and half of them still follow hunters and gatherers lifestyle. That means they go and hunt for food. They don't do any job including agriculture. So now I am coming back to the question which I left, the admixture which took place between ASI and ANI. We could not address that, and we tried to address when this admixture must have took place. That's one of the important questions at the time. To address that, again, we used large number of population groups from south and large number from the north. As I mentioned in the karyotype that every generation we inherit 50% of the genome from father, 50% of the mother, right? So by looking at that, and when you expect that there's a recombination between the homologous chromosome, there's a way of estimating the age of the admixture, and we came to know that these two founding populations, ancestral South Indian and ancestral North Indians, have initially gave rise to many population groups independently. Right? Just imagine that several thousand years back, some populations settled in a jungle. Okay? They finished food in that region. Some group thought, okay, so they have to go and hunt for food in different places. So they migrated to other places. And from there they migrated to another place, started agriculture or domesticating animals and so on. So that's how several groups have diverged. Then after that, what happened for the last 2,000 to 4,000 years, there are groups which have admixed. The ASI and ANI have admixed. The color indicates that the genome have admixed. Right? So after admixing, every single population then started maintaining their endogamy. Endogamy means marrying within the community, either caste or tribe or religious group, any kind. They maintain the endogamy. That means in a genome, any organism you take, including human, if the population is isolated, naturally there will be a mutation in the genome, and some mutation arose in one particular population, not necessarily the same mutation you would expect in the other population. When the mutation exists, when the population size increases, is going to be restricted only in that population. Similarly in the other population. So now, because of the endogamy practice marrying within the group, and whatever the mutation accumulated in each population remains only in that population, that makes every population unique. Okay. And this endogamy practice, which happened 2000 years back, is much before the caste system. You know that caste system arose around 1500 years. That must have put more pressure on the populations to maintain the endogamy. Okay. Now what I am going to talk to you is that what is the impact of the endogamy when population started maintaining or marrying within the group, what kind of impact that would have on our health and disease. To understand that, we thought we should study large number of groups. So in this study, we went out of India, not only restricting only within Indian population, went to other South Asian countries. If you include other South Asian countries, then the population size is around 5,000, which is again largest number of diversity anywhere in the world. That includes population from Sri Lanka, Pakistan, Nepal, and all other South Asian countries. The reason why we went to other population also is because of our other studies. In one of the studies, where they have shown that all the South Asian groups, as you see there in the structure analysis, the dark green, which is restricted only within the South Asia. Right? Then the light green extended beyond South Asia. But when we study this component, the genome which is restricted only in South Asia, we found that at least 50% of the genome has gone from India to other countries. That means India genetically is a mother for other South Asian countries. Okay? So this is one evidence. The second evidence, we also look at many diseases, including disease called cardiomyopathy. This is one of the heart muscle disease, and there is a mutation in this particular gene called myosin binding protein C3. This is a 25 base per deletion, which actually leads to disease called cardiomyopathy. It's a heart muscle disease. There are three different kinds of cardiomyopathy. One is dilated cardiomyopathy, where the heart enlarges beyond the normal size. Second one is hypertrophic cardiomyopathy, where the heart muscle becomes very thick, as you see here. Then the restrictive cardiomyopathy, where the heart muscle becomes very stiff. So because of this condition, suddenly the heart stops its function, therefore sudden cardiac arrest. When we found this mutation, if the mutation is in homozygous condition, that means both the copy of the genes, as I mentioned that there are 23 pairs of the chromosomes, 46 chromosomes. If the mutation is present on both the chromosome pair, it's called as homozygous, then the individual died at very early age. By looking at the post-mortem tissue of the children who died at very early age, we found that the mutation is present in the homozygous condition. But in the heterozygous condition, the individual live up to 40 or 45 years without any symptoms. But suddenly they collapse. Particularly the athletes, if they have the mutation, and if they are running or they playing or cycling, suddenly they collapse. Okay, such a kind of disease it is. And coming to the context, what I am showing here is that when we try to look for the mutation within India, we found the mutation is present all over India except Northeast and Andaman and Nicobar Islands in about 4.5% in a heterozygous condition. That means people can survive up to 40 or 45 years without any problem. Then we went on to screen the population throughout the world to see how the mutation is present in the entire world. Then we found that the mutation is restricted to only South Asian country but not in other country. The one which is in the yellow color are the one who carry the mutation. That means this mutation is South Asian specific. And in fact, we did a lot of other studies to find out when this mutation arose. It arose about 30,000 years back. Okay, now you can correlate with the population study. There's a two founding population. There's an admixture. Doing this admixture got well spread across India. And also there was a migration to other South Asian countries. During that time, it got well spread. So now with the population background, we could clearly understand when this mutation arose, how it got spread. Okay. So with this background, we thought we have to look at the populations from South Asian countries. Now we took 2,800 individuals from 275 population from South Asia. And the dots indicates where the samples have been collected, and the color indicates different linguistic families, including Andamanis, Nihalis, and few more linguistic groups. That's isolated linguistic groups. So what we came to conclusion by studying is that there's a lot of opportunity for us to identify a population specific rare diseases, initially to mutation and also the rare disease. Okay, so that's one of the again important findings, that we did not look for any disease causing gene. We not even know that the populations are having any population specific disease, but genetically we identified, yes, there could be a population specific mutation, particularly the recessive mutation in majority of the population in South Asia. So how we came to the conclusion is that this is called as IBD score, identity by descent. So every single line represents one population. Okay, the length of the IBD shows that within the population, take any population and take two unrelated individuals. When you compare the sequence, if they are really unrelated, then you should see lot more variation between those individuals. But if they say they are unrelated, but if you see the sequence is identical, that means today they are unrelated, but several generations back they must have related. So they must have come from a common ancestor. Okay. The problem is, because the population practice endogamy, marrying within the population, if the mutation arose, if the mutation is dominant, that means even if the mutation exists in one other pair, it will express the disease. But here what you are talking is the recessive mutation. If the recessive mutation exists only on both the chromosomes, only it expresses the disease. If it is present in only one chromosome, call it as heterozygous, then it will not express the disease. So the mutation can spread across the population. The frequency will increase because it's not going to cause any disease in the heterozygous condition. Then to some point of time, when the mutation is well spread, then there's a possibility that two heterozygous individuals, the boy and girl who is going to marry, will carry the mutation in the heterozygous condition. If they marry, there's a possibility that they contribute the abnormal chromosome to the next generation, therefore there's a disease. That we'll see in, I'll explain much better way. So here, by looking at this IBD, large number of population in India have high IBD score. That means there's a possibility of population specific recessive mutation and the recessive disease in large number of population. I put two population here. One is Ashkenazi Jewish and Finnish population. They have very tiny IBD, the red color one, compared to large number of Indian population. But you can imagine with this small IBD, there have been large number of disease have been mapped. Bloom syndrome, cancer, right? Caravan disease and many cystic fibrosis. There are large number of disease in the Ashkenazi Jewish. And these are the genes. And if you see this, these are all autosomal, most of them are autosomal recessive disease. Okay. Similarly, Finnish population, there are large number of disease. And you can imagine with this small IBD itself, they have large number of such disease. What would be the consequences of large number of population who have several fold higher IBD score? Only thing is that we don't have proper data and medical record that if you go to village, if somebody has the disease in a 1 or 2 families, and they are not going to report to the medical community, so that remains there, right? Like that, I don't know how many such cases exist in India. To explain that how the mutation goes generation after generation, you can see this pedigree. I hope that children have studied, the pedigree, right? If you put circle this indicates female. If you put square, male, and if you put line here, that means these two are wife and husband, married. And if you put line down, these two are their offspring. Two daughters. If there is a son, then again if you extend it and put square, there is a son. Right? It goes. So imagine that the mutation is in one of this chromosome, but she is absolutely fine. That means it's heterozygous mutation. Then it goes to the next generation in again one chromosome. No problem, because in the heterozygous condition, both of them marry somebody from outside. They don't have that mutation. Again, no problem, it goes in the heterozygous condition at all in many generations. Right? After several generations, they must have lost their relationship. Okay? Even if you go to some village, remote place, there may be 100 families. Now, two families, they may say that they are unrelated, but actually you can connect. They all must have come from a common ancestor. They must be carrying the mutation. So in that condition, if you see these two individuals now ready to marry, they are in the population, they wanted to marry. Now you see that both of them carry the mutation in heterozygous condition, but they are absolutely fine. But if they marry, statistically you can say there's 1/4 of chance that they contribute the abnormal chromosome to the next generation, when both the copy of the chromosome carrying the mutation will lead to the disease. There are several examples. To test this hypothesis, we took a disease called progressive pseudoheromatic dysplasia from again South India. There are 30 families. We have studied all the 30 families, and here you can see that the disease is very severe, and there is a swelling in the joints, all the joints, right, with the severe pain. And we found the mutation in 6 individuals, 6 patients. Their parents are absolutely fine, no phenotypic feature at all, right? But they are carrying the mutation on other chromosomes. And 6 of them, then we try to see whether all the 6 are relative or not. Although they say that the parents are unrelated, right? Then we sequence the flanking region of the mutation to see whether all the 6 are identical sequence. There is no difference at all. Then we found 5 out of the 6, although they say the non-consanguineous, not related, but they have identical sequence, same haplotype, suggesting that today they are unrelated, but several generations back they are related. Okay. Not only this, there are several examples. This is another disease called mitochondrial disorders. Mitochondria I explained some time back. It has its own DNA, and there is a nuclear gene called polymerase gamma. Polymerase is an enzyme which is essential for DNA replication. You must have studied, right? Here the nuclear gene, there is a single base mutation. C is the wild type, normal. T is the mutant. This is called heterozygous. I told you, 2 copies of chromosome. When you sequence, the sequence is identical. That means both the chromosomes have the same sequence. Here, 1 chromosome is normal, 1 chromosome is abnormal. Okay, similarly here, what you see, father who is 45 years, mother is 39 years with the heterozygous mutation. What you see is C and D. Okay, but still they are perfect because only 1 chromosome is affected. But after the marriage, you can see 2 of their children, of 3, have with the homozygous mutation, like T. Father contributed T, mother contributed T, same way here. So unfortunately, both the children have died. It's a very, very early death. What is the problem here is encephalopathy, seizures, stroke-like episode, and premature death. And when you look at the mitochondrial level, because of that mutation, it introduces large number of mitochondrial multiple deletions, mitochondrial DNA deletion. And these are some of the markers which show that accumulation of mitochondria, abnormal mitochondria, and the lack of COX-negative fiber and so on. But here what I am showing is that this is typically it's following what was our observation. Okay. Same way in this family also. But what is important here is because we know the cause, one single base mutation leading to so much abnormality and premature death. But this boy with heterozygous mutation, when he grows and wanted to marry somebody from within the relatives, we can just test for this particular mutation. It costs only few hundred rupees, 200 to 300 rupees. But that can save the entire future of this family. Okay, that's the one. But similarly, what I wanted to highlight is what the Ashkenazi Jewish practice, because they have large number of patients, large number of diseases have been identified. So they have organization called DO ratio. So this organization goes to school, collects sample from the children, sequence them and put it in their database. But when they grow 20 plus, they wanted to marry, boy identify the girl or girl identify the boy, they wanted to marry. They take out the sequence and compare the sequence and see whether both of them are carrying the same mutation or not. Then they advise either not to marry, or even if you marry, you have to look for that particular mutation in the fetus. Then if the fetus carry homozygous mutation, then you have to decide whether you want to terminate or continue. If you continue, what would be the consequence and so on. So by practicing this way, in fact they eliminated a couple of diseases from the community itself. So that is what is required. Country like India, where we predicted the large number of population in India are prone for population specific diseases. So in that line, when you are looking at some of the rare disease, this is one of the rare disease that held its epidermolysis bullosa. So this disease, where you see the lot of lesions on the skin, oral mucosis, congenital heart disease. Again, the children die at a very early age. So what we found that in a southern India, there are many individuals, family in a small villages. There are 22 villages, and most of them carry the mutation. It's a similar trend. The mutation 11 base pair deletion in LAMB3, both mother and father, they are heterozygous, absolutely no problem. One chromosome is normal, one chromosome with the deletion, and they are passing on to that, both of them, therefore homozygous mutation, and therefore premature death because of all this issue. So not only this, there are families. Here there are 5 families. Wherever there is a black color, that means these are the children who have died. And wherever this red mark, these are the sample we have analyzed and found the mutation in heterozygous condition. Clearly it is following that trend. So now we can screen the entire population and we can give appropriate genetic counseling. Therefore, we can avoid this kind of children in the future. So this is one way where we know the disease, then try to look for mutation which causes the disease. The second one, there are several other population. This is called Hadikudu disease. There's a place in Karnataka, place called Karnataka, that's Hadikudu village, and they named the disease, putting their name because it's a place where they are very short stature with a lot of bone abnormality. Right? That's restricted to only in that area. Similarly, here there's a place called Udanam in Andhra Pradesh, called as Udanam kidney disease. A lot of studies has been done. Some study has been done here also. Looks like whether the food habit changes this. Okay, they are undergoing the dialysis. And there are many more diseases like cardiomyopathy, which I talked. It's a region in Telangana, Andhra Pradesh. One particular community called Raju is a predominant of cardiomyopathy. Similarly, many more diseases like arthritis, one particular region. This alkaptonuria and gypsies in Tamil Nadu. Vaishyas have problem of pharmacogenomics. There's a pseudocholinesterase deficiency. So they're all population specific. Earlier I showed the children dying. These are well-known population specific disease. We are also looking at population with high IVD score, where we don't have any clue at all what kind of disease they have. By doing this, probably the next few years, we can able to identify the mutation which is responsible for the disease and continue giving appropriate counseling. Therefore, after a few generations, we can avoid such kind of disease in future. So that's what is the future plan. With that, I would like to thank all my collaborators, colleagues, right from CCMB Recognition, all my students. There are a large number of collaborators within country and also outside the country. Otherwise, this is not possible to do this kind of studies. And the funding has come from CSIR, UK India Education Research Initiative, a grant, Department of Biotechnology, Department of Science and Technology, and Medical Research Council at London. And thank you very much. I hope that you all had. I'll be happy to address if you have any question.
Yeah, if anybody has a question, please raise your hand.
And you can think over a lot of opportunity for you people to study genetic genomics, right? There are a lot of population can be studied at disease level and molecular level, how to prevent. No, there are many aspects. So as children, you can think right now.
I myself have one question, sir. May I ask?
Yeah, please.
So, this is related to any nutritional disorders which you think have genetic component, which are well known in the area of genetics.
Yeah, there are many.
Yes, exactly.
Yeah, there are many diseases have been identified. Even if you take like vitamin D deficiency, it has a genetic component, right? Vitamin B12, many more. All of them, I think as a geneticist, I believe that every aspect if you take, there's a genetic component in that.
The next question then is whether we should have population-specific standards for this new various micronutrient deficiencies.
Yeah. One can do that. We have to do systematic study to have that.
Thank you.
Yeah.
Thank you very much, Dr. Thangaraj, for that very interesting and fascinating talk on population genetics. And I'm sure the audience is equally fascinated, and it has inspired them to, take up science and genetics as a career. I would now request Dr. Ahmed Ibrahim to felicitate our guest speaker with a shawl and a memento.
But now he's an administrator, you can understand his busy schedule. Thank you once again, sir, for sparing your time. He was trying to see whether he can take it on online, but I said for the benefit of the students, please come, and he has come. Thank you so much, sir. And now kids can announce it.
Sir, Rajesh, sir. Rajesh, sir. Next you may go, please you can go from the back door in batches. As we can open it. School wise.